We're here today with Ask AI podcast host Jaxson Khan, and he has some exciting news about his affiliation with the Ask AI podcast, and we're here to chat with him about that. Jackson.
Awesome. Thanks so much, Melissa. It's been an awesome few years working on the podcast, and I am a little bit bittersweet moment, but I will be moving on, and in fact, handing it over to you, which I'm extremely excited about. And with that, I'll simply say thank you. Thank you to everyone, and also thank you to
Thank
all your
you.
listeners. I've really enjoyed being a part of this, and I'm grateful. And I wish everyone the best. I'll keep on following now. I'll keep on listening. And with that, I've been your host, Jaxson Khan. Take care.
Welcome to Ask AI, the podcast that brings you insightful interviews and news from the world of Canadian artificial intelligence. This episode is sponsored by Microsoft Canada. Microsoft is committed to building trusted and responsible AI systems. To learn more, go to microsoft.com/AI and check out their free AI business school to start building intelligence into your solutions today. We're also sponsored by Cinchi, the global leader in data fabric technology. Visit Cinchi.com to learn how to eliminate integration and turbocharge your AI transformation.
Hi there, artificial intelligence enthusiasts. Welcome to this edition of the Ask AI podcast. I'm your host, Jaxson Khan. On today's show, we have Dr. Samuel Aparicio. Professor Aparicio is the Nan and Lorraine Robertson Chair in Breast Cancer Research at UBC, is the head of the BC Cancer Research Institute Department of Breast and Molecular Oncology in Vancouver, Canada, and also a fellow of the Royal Society of Canada. He has most recently conducted foundational work on methods for studying the evolution of human cancers using next-generation sequencing approaches, as well as single-cell sequencing methods. Dr. Aparicio is also working to develop quantitative measures of clonal fitness in patients, including methods for single-cell genome sequencing and patient-derived xenograft models of human cancer. If you understood all of that, much kudos for me. But nevertheless, I'm extremely excited to be speaking with you today, Samuel, and to learn much more about the work that you are doing. And I would want to welcome you to the Ask AI podcast.
Well, thanks for the generous introduction, and it's great to be here.
Fantastic. Well, to start things off, why don't we start with some of the simple questions? How did you get involved with artificial intelligence in your work? And perhaps you could give us a bit of background on your work in general.
Yeah, thanks. That's a great question. And in fact, my interest in AI and machine learning, I guess what people would call statistical machine learning, goes back quite a long way, way back into the start of my research career when I got very interested in the way that neural nets function. And this is going back in time a bit now, sort of into the early 1990s when computer scientists were beginning to learn about the ways that you could apply neural nets and statistical machine learning to problems where it was necessary to look for patterns in data or to understand the behavior of very complex systems. And that seemed to me like a tremendously powerful approach to bring into biology. And so I kind of got interested in the field then. And, fast forward to the last 10, 15 years where my research group has been trying to decode cancer. We've realized that the very powerful techniques for finding patterns and studying the way that dynamical systems, things that change over time, how that works, there are very profound applications now of modern AI and statistical machine learning.
That's remarkable to hear about some of those early findings about what might be possible. Are you able to provide us with perhaps I don't want to say it too broad of an update, but that in terms of the state of global cancer research and where we are today, have we been able to achieve some of the big milestones that we've been watching to over the years as a global community? I'm just curious about where we stand today.
Yeah, great question. And I think we're often asked sort of how are we doing in the global war against cancer? I think the easy way to say it is that we are certainly preventing, treating more cancers, and also helping patients with advanced cancer live longer and with less side effects. And it's actually sometimes instructive to kind of stop and look back the road that's been traveled, because cancer treatments and cancer control in general was much less effective 30 years ago. I can give you a kind of quantitative example. There is just we know that for women with breast cancer in the 1970s, early 1980s, the average survival after a diagnosis of breast cancer was around 50 or 60% at 5 years, and that's now been pushed up to around in Canada, US, Europe would be around 92%. So there's been a big impact in terms of the survivability of cancer as well as the treatments. Now, having said that, we've had a lot more success with certain cancer types than with others, and so we're focusing a lot of attention on what are called hard to treat or hard to manage cancers.
Mhm. And that's just astounding to hear, the difference in survivability now. That really is remarkable, and I hope that you and so many of your colleagues, collaborators around the globe are extremely proud of that. I suppose what I'm curious to learn more about is, as we're starting to embark on new areas of research, or to improve those rates even more, what role does AI play in advancing cancer research today?
Yeah, it's playing an increasing role, and the way that we think about AI has to do with the role of the measurements that we make and the ability to discover patterns in data, and also to search for hypotheses with a sort of an AI basis. Um, and I could, mention a few examples here. Um, the role of measurements is key in cancer, and what I mean by measurements are measurements in trying to understand how cancers arise and how to treat them come from all manner of data that we collect. Some of it comes from studying the way that the genomes of our cells get altered in cancer. So the genomics of cancer has driven a lot of understanding. But there are other forms of data that we collect about cancer, such as the patterns and incidence of cancer across the world in different populations. We learn about symptoms and side effects, the effects of drugs. There's now, since about the mid 1980s, a huge rise in what is called cross-sectional imaging. That is, people would know that as CT scanning or MRI scanning, which oncologists often perform to either look for cancer or to manage a cancer. And all of these different forms of measurement, be it sort of epidemiological, behavioral, molecular imaging, generate data that we use to understand how cancers arise and how we might set about either preventing them, diagnosing them earlier, or treating and managing them. And that it's a huge puzzle to distill patterns out of that data, and AI plays a very natural role there, because a lot of the data we're now collecting is increasingly already digital at source, and so it's very amenable to computation.
Very, very, very interesting to learn it. And I guess one thing I want to dive into a little more is specifically what type of data is fueling your research, and curious how that where you find that data, how it's managed, governed.
Yeah, great question. So where the kind of research that we do, when people ask me sort of what do we do in cancer, I guess the shortest way of saying it in a contemporaneous thing is that we fight variants in cancer. I think everyone understands what a variant is these days, with the pandemic. But this process that organisms can mutate their genomes. So genomes, when they replicate, when we reproduce cells, they can accumulate spelling mistakes in the DNA, and most of those are inconsequential. But sometimes one of those mutations will have a consequence, and that consequence leads to altered behavior of cells, and there you're really describing cancer. And so we spend a lot of time trying to decode the genomes of human cancer in the context of treatment, or in the context of the origins of cancer for people who may be at high risk. And we use all of that genomic information together with measurements that we take over time to try and learn about the way that drugs may work, or the way that they may stop working, or to suggest new ways in which we might either prevent or treat cancer. And the data has a huge dimensionality to it. We're taking literally millions of measurements on every cell, and there are millions of cells and hundreds or thousands of patients. And so when you multiply all those dimensions, you can see that we end up with a very, what's called a very high dimensional space.
Wow, that's incredible to hear. How sort of the scope of the data that you're reviewing and analyzing and working with, it just continues to expand in the various dimensions and aspects of it. I suppose if you could wave a wand, is there a data set out there that your team would love to have and work with?
We would love to collect or have data corresponding to the genetic decoding of every patient's cancer, plus access to the kind of the non-molecular, non-lab medical data. So when patients enter the system, either as an individual who might be, say, at risk of cancer and is being screened for cancer, or who may have developed a cancer and is being treated for a cancer, all manner of information is collected about as part of management. The normal management of a patient's course would be collected about what drugs were they treated with, or how did they respond, what was this or that image, what did it look like. And we seek access to this information, but it's still, I would say, quite hard for us, very hard for us to really collect this in a very systematic way, for a whole variety of reasons. But if I could wave a magic wand and kind of get access to all of that, it would just be tremendous.
I can imagine it. And just to walk me through for a moment. So when you say the genetic decoding of their respective cancers, what does that particularly involve? Like how does that get done, or how does that happen in the first place?
So over the last 10 years, there's been I would fairly describe it as a revolution in our ability to determine the genetic sequence, the sequence of DNA in organisms.
Mm-hmm.
And these were technology advancements that started around about the early 2000s and really came of age around 2009, 2010, when our capacity to decode, to take any particular organism, be it human, a bacterium, a virus, and learn about the base by base nucleotide structure of the organism, was transformed orders of magnitude into something that I never thought I would see in my professional lifetime, which is the ability to routinely determine the DNA base sequence of every human genome that we would ever want to study. And that has transformed our ability to study all manner of diseases.
And if I'm following you, so some of the applications that I might be familiar with, for example, might be the consumer friendly ones like 23andMe, for example. That's my understanding. But then also, frequently, is that able to be applied, for example, if someone is in the late stages of cancer, or potentially has passed away from cancer, is that able to be done on their behalf to decode their cancer?
Yeah. In fact, and we do this all the time now. So
Okay.
the 23andMe examples are actually quite limited in scope, and have to do with our constitutive DNA and the way that variation in our genes might influence diseases that we might be prone to. But it goes a lot beyond that, because cancer is a disease where what we call the somatic genome, that is the genome of cells that make up parts of tissues of our body that don't necessarily get transmitted to the next generation, but they nevertheless, those genomes get mutated you can end up with a cancer. And so we've begun to learn how to interpret the patterns of mutation in all sorts of ways that will tell us about how a cancer might respond or what caused it in the first place. And indeed, eventually, for some individuals, that there could be a family risk, for example.
Well, and I was just trying to get a sense of how it worked and even what the scope of this type of decoding and analysis is. Would you say, for example, in your work with BC Cancer, for example, is that something that's happening to 5% of patients, 10%, 20? Like, is there a rough scale or percentage of people that's happening to right now?
So there's a small proportion of patients that we are able to enroll in research studies and decode
Okay.
the entire genome of their cancers. There's an increasing proportion of patients who get as a matter of routine care a sort of limited look into the genome, which is also done through genetic sequencing. It's called targeted sequencing, and that's used in certain cancers now to actually look for indications that a cancer might respond to a particular drug. So that's beginning to be used in a limited way, and those applications are going to rise. The current diagnostic applications are still looking at small parts of the genome because those are the parts that are interpretable at the moment. But it's becoming rapidly obvious that by sequencing the whole genome, we're going to be able to do things like have more sensitive tests to find out whether cancer is still present in someone's body after a treatment and what to do about it. And we're learning about those things by making applications of AI and machine learning to pattern discovery to give us more sensitive tests for managing cancer.
Well, so that's really, really interesting, important information to know. And so I suppose this is my my limited knowledge and brain trying to now see if I can connect this to some other contemporary examples. But everyone now is obviously, as you mentioned, more familiar with the term variant, for example, and also the term we hear about all the time is mRNA. It is part of this genetic decoding work tied to that process, or is there some sort of coordination with mRNA vaccines or work or future solutions that does tie into some of the work you're doing? For example, I even heard the other day there's mRNA now being applied to an HIV vaccine, but is there any application of that to the type of cancer work you're doing now?
Yeah, that's another great question, actually. And one of the things we've learned about cancers is that the mutations that happen in cancer are something that could in principle be tackled by enhancing the way the immune system works. And people have approached that question in a number of ways, but one of the obvious ways to think about it will be to say, well, could we create a vaccine that helps the body's immune system react against the mutations that are present in a cancer? And that is very much linked to our ability to decode the genomes of cancer. And so in fact, the companies that have sort of become famous for the mRNA vaccines during COVID, their primary programs before they got into COVID were really to do with creating cancer vaccines. It was exactly that application that they were chasing, and the arrival of a new virus it was an obvious thing to do to sort of divert the technology and apply it that way. But this is something that the field is chasing down for sure.
Wow, I had no idea. This is so exciting. Are you able to describe to our listeners some of the other key projects you're working on at BC Cancer and your work?
Well, as I mentioned, we're very interested in what is it that causes cancers, and also how can we identify the kind of vulnerabilities that cancers might have. And what we're very excited about at the moment is that through the work that we've done looking at the structure of genomes using this genomic sequencing technology that's around, coupled with the statistical machine learning and AI that's been applied, we've begun to find patterns of breakage in DNA that suggest certain mechanisms. They have to do with the way that chromosomes in our genomes are kept together. And we think we see a way to begin to exploit some of this in order to generate kind of more shots on goal, so new therapies that we might find. And I'd say the most exciting part about this is that we're beginning to start to make sort of causal inferences about and reason about the way that certain genes and structures in the genome might contribute to this with the assistance of methods that come from AI, and also with the observation that because we can go in now and sample the evolution of cancers, we can study over a period of time just how these changes are happening, and that turns out to be tremendously powerful. So we're engaged right now trying to develop new classes of drug based on this kind of measurement and pattern discovery that's been AI assisted. And that's, our work has been very focused in breast and ovarian cancers, but we're beginning to learn that we're seeing similar patterns turning up now in some childhood cancers and pediatric cancers and in other cancer types, where because we're able to look using the AI methods that have been developed, we can see patterns emerging that are sort of helping us reach beyond the specific disease we've been studying.
That's really, really fascinating. And I was actually curious, for example, what happens if the AI assisted process discovers a pattern that perhaps you weren't looking for or were focused on prior, and was curious how that might tie into how you work within the research ecosystem and potentially engage other partners. So for example, like if you do discover a pattern that maybe your team is not particularly focused on, but do want to alert others in the ecosystem, how do you handle those types of insights?
Yeah, the way we're all talking to each other all the time. So,
Mhm.
the first thing that might happen is I might phone up my friend in Los Angeles or in the UK or something and say, "Hey, we just saw this thing, and I know you work on this. Like, can we talk?" So that would be the first thing. And one good thing that has happened through the pandemic is that we've all learned how we can do that virtually much more easily than sort of waiting to go to a physical meeting. We like to go to physical meetings. Lots of great discussion happens when people are in person, but that kind of communication happens. And also, we publish all our research findings. Like eventually, the
Mhm.
data, the findings, and everything go out into the public domain. And one example of that is one of the very first things we did in the program in Vancouver back in around 2009 was we conducted a really large study at the time of the genomic structure of 2,000 patients with breast cancer. It's still probably the largest study of its kind. Um, when we did that study, the initial study, we said we're going to put all of the data into the public domain. So that's the genomic data, the associated information about outcomes, how did the patients respond to treatments and so on. We put all of that information in the public domain, and that data set has been reused by now over 450 groups and institutes worldwide. And that publication has attracted a lot of citations as a result, and it's because the data set is reusable, so people can go off and discover their own facets of what's there.
That's wonderful. Huge fan of open data and access and seeing what might come about from interested parties and people who want to work on this type of work. That's fantastic. Um, and more broadly, sort of beyond the work, the research you're doing right now in this moment, what is most exciting for you about artificial intelligence right now?
Well, I'm just fascinated at how the field is developing. It seems in a very multidisciplinary way. So the obviously it's a field that's been driven heavily by in a fundamental way by sort of statistical machine learning and computer science, but the influences, sort of the technical influences that seem to be creeping in from physics and other sort of disciplines seem quite fascinating and are moving the field forward. And I'm seeing advances in the ability to pull out patterns, model complex systems. I'm particularly excited about maybe the ability to reason causally about how one thing influences another. And that's the kind of thing where I think if we could scale causal inference in a robust way, we would really move forward a lot of biomedical discovery, because it would help augment the way that we look for hypotheses, which in the end we have to kind of go off and physically test things in the field that we work in. But our data sets are becoming so complex and high dimensional that pretty much the only way you control a data set to sort of look for a hypothesis or to try to validate a hypothesis are with these sophisticated tools.
I can certainly see some parts of that. I mean, I do wonder though. I mean, is there a risk to this? As you said, potentially we can only find some answers using these types of tools. But I guess who controls those tools, or who ultimately publishes about them? That's maybe sometimes what I worry about. Maybe this is thinking too outside of my own my own lane, but I know what I've often heard about in sort of modern science is the replication crisis. And so what if only some people can use and really understand these tools, and we're not able to replicate findings and really verify their validity?
Yeah, that's another great area to consider. I think what we believe in is that when you fully expose data and you also fully expose the computational and statistical methods,
Mm-hmm.
then what happens over time is that people will either some people will try to reproduce and they can't, or they try to reproduce and they can, but they find something subtly different. It's I think putting data and methodology into the public domain is one of the most fundamental aspects of science, because if you can't have people who are kind of sort of not biased by the original discovery process and the excitement that goes with it and so on, if you can't have people kind of reproduce or try to reuse things, then it kind of that's not a great foundation on which to build things. And in general, I'd say actually there's been a very positive move in that direction. I think academic journals now pretty much insist on disclosure of data and methods. The field hasn't really gone far enough, I think, in terms of making that accessible. It's still stuff gets out there, but it can be quite complicated to untangle how everything it fits together, because some of these data sets are extremely complex, and so learning about all of the features of the data set may not yet be a kind of a fully it's not as efficient and transparent as it should be, but the field certainly works in that direction.
Well, yeah, let's let's hope that it continues to evolve that way and have more open data, have it be accessible, have those models be available for folks to evaluate. I think, as we're kind of in this interesting moment in the world where there has been a crisis of trust in institutions and establishment, I think that's the kind of thing that potentially could result in more trust and more access. So I fully support that. Coming up on our time here, but I did want to ask you perhaps the question that we must is, and what maybe on the minds of many, is Doctor, do you ever think that there will be a cure for cancer?
The shortest way to say that is that we will never prevent everyone from developing cancer. I don't think that that's likely. I do think that it's likely that we will reach a future where if we don't cure cancers, they become more like a chronic disease. And that, in fact, we are, in the sense that people commonly understand the word, we are curing more people. We're pulling more people out of the river when they fall in. We're not going to stop everybody from falling into the river, but we are pulling out more people, and we're also helping more people live meaningful and symptom-free lives with cancer as a chronic disease. So that, we're getting there.
Well, I think that's a really wonderful analogy and way to leave it off. I think it's a very positive message. I'm extremely grateful to you for making time to share more about your work, more about the advances that are being done in cancer research using AI assistance. And I know I probably speak on behalf of a lot of folks that you've positively impacted their lives through your work. So thank you so much, Dr. Aparicio.
Well, thank you. It's been a great discussion.
Fantastic. Well, thank you again. And I'd like to also thank our series sponsors, including Microsoft Canada. Microsoft hosts the free AI Business School, where you can learn how to drive effective business impact by creating an AI strategy, enabling an AI-ready culture, and innovating responsibly. Also, thanks to Sinchi for sponsoring the Ask AI podcast. Sinchi is a global leader in a data-aware platform technology that makes data integration obsolete, restores data control, and accelerates hundreds of transformation projects. And last, we're also sponsored by ScriptSwap, the AI-powered jargon buster that simplifies business contracts with simple language and formatting recommendations that everybody can understand. And don't forget, if you are a team working in Canada and you want to share an update with the Ask AI audience, visit our website at askai.org and click on Team Check-ins. We'll be happy to speak with you. Thank you so much again. We'd love to hear from you. I've been your host, Jaxson Khan. Thank you again, Doctor. Thank you to all of our listeners.
Thanks for listening to the Ask AI podcast. The sponsors of this episode were Microsoft Canada, producers of the free AI Business School, and Sinchi, the dataware platform that makes integration obsolete. The series producer was Chris McClellan. The series editor was James Fajardo. Original music was provided by Mike Letourneau. To learn how to be featured on our podcast and get information about sponsorship and volunteering opportunities, please visit our website at askai.org, send us an email to info@askai.org, or talk to our bot by visiting askai.org/chatbot.